and some invited subject matter experts. PhilSci-Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment. And the best part? It's absolutely free. Visit us at philscihub.com and be part of this learning journey. Let's learn and succeed together here at PhilSci-Hub Research University. Welcome to PhilSci-Hub Research University, your ultimate guide on your next research project. We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the PhilSci-Hub team of research scientists and some invited subject matter experts. PhilSci-Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment. And the best part? It's absolutely free. Visit us at philscihub.com and be part of this learning journey. Let's learn and succeed together here at PhilSci-Hub Research University. Welcome to PhilSci-Hub Research University, your ultimate guide on your next research project. We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the PhilSci-Hub team of research scientists and some invited subject matter experts. PhilSci-Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment. And the best part? It's absolutely free. Visit us at philscihub.com and be part of this learning journey. Let's learn and succeed together here at PhilSci-Hub Research University.
Mabuhay! Welcome to the Filipino Science Hub. Thank you for your continued support to this online platform. To our new participants, thank you for checking us out today, and we would like to officially welcome you to the PhilSci-Hub family. Before we start the webinar, I would like to share some house rules in order to facilitate a smooth session. For our Zoom participants, we would appreciate it if you could turn your audio and video cameras off during the webinar. If you have questions or comments, feel free to type them on our Zoom chat box or YouTube live comments, and we will try to accommodate as much questions as possible. After the talk, we will have a 30-minute Q&A where the Zoom participants will have the opportunity to ask their questions live. For those who would like to receive a certificate of participation, a Google form link will be posted on our Zoom chat box, YouTube live comments, and YouTube video description towards the end of the webinar. Again, welcome to all our Zoom participants and YouTube live viewers. For those who are new and may not be familiar with who we are and what we do, allow me to share our mission and vision in the next few minutes. Filipino Science Hub is an online platform founded by our CEO, Filipino scientist, Dr. Jeffrey Camacho-Bonquin back in 2012. Currently, we are a registered nonprofit organization based in Houston, Texas, United States. Our mission is to promote STEM education and the culture of research among students and teachers in the Philippines and abroad. Our current global leadership team is comprised of volunteer Filipino scientists, seasoned STEM educators, and business executives across the globe. Our vision is to see a technology and innovation-driven Philippines, and we believe that this can be achieved by creating a new generation of well-rounded Filipino STEM enthusiasts. The vision is being carried out via our two major programs, the PhilSci-Hub Ed program and the PhilSci-Hub Research University. Through PhilSci-Hub Ed, we are able to empower and support our teachers by strengthening STEM fundamentals education. We believe that this is important because the more equipped our STEM educators are, the more they can inspire students to pursue a career in science, technology, engineering, and mathematics. The other major program that drives our vision forward is PhilSci Research University. This is where we provide tools for fundamentals of research and bring practicing scientists closer to the educational sector. Our official website is www.philsci.com. This is where you can access all our materials such as webinars, tutorials, modules, virtual lab, and features, all of which are free. We are also present in several social media platforms, so follow us wherever you are most active on to get updated with our web events. Again, thank you for being with us this morning. To officially start our webinar, I would like to turn the virtual floor over to our host today.
Alright. Magandang umaga po sa inyong lahat sa Pilipinas. I am Jeffrey Bunkin. I am a Filipino scientist based here in Houston, Texas, and I am the founder of the Filipino Science Hub. So, at this point, unang-una po, gusto namin magpasalamat sa inyong lahat for joining us today, for spending your Saturday morning with us, with the Filipino Science Hub. Today is a very important day because this is the 109th web event that we have hosted since we started our training series in May of 2020. So we've been doing this for about 4.5 years now. We're happy to be able to bring Filipino scientists and international experts in different fields from all over the world closer to our Filipino students and educators. And for our 109th offering, we're happy to share with you um, a very timely and relevant topic. So this is actually it's for today. Like our talk is gonna focus on fungi. So fungi as a growing threat to human health, and it's a question of do we continue to ignore them? And so today we're also graced by an internationally renowned Filipino clinical mycologist, Dr. Connie Hibas. And Dr. Connie Hibas is also the uh, head or the chief of biology in Filipino Science Hub. So before we hand the floor over to you, let me do the honor of introducing Dr. Hibas to each and every one of you. So Dr. Hibas, as I said, is a Filipino mycologist. So she uh, uh, earned her bachelor's in medical technology degree from Silliman University. She then went on to earn her master's in veterinary medicine, specializing in veterinary microbiology at the University of the Philippines at Los Baños. And then after that, she actually pursued her Ph.D. in biological sciences, focusing or majoring in fungal systematics at the University of Alberta in Alberta, Canada, where we met. So, as I mentioned, so at the moment, Dr. Hibas is a clinical mycologist and clinical research project manager at the Fungus Testing Laboratory at the Department of Pathology and Laboratory Medicine of the University of Texas Health at San Antonio, Texas. Before that, she was a curator at the University of Alberta Microfungus Collection and Herbarium in Canada. She was also a research fellow at the Japan Collection of Microorganisms at the University of Tokyo in Japan. And before that, she also worked in the Philippines as a university researcher in the Philippine National Collection of Microorganisms, Biotech at UP Los Baños. So that's her educational and work background. But these are some of the highlights of her career as a mycologist. So, Dr. Hibas has discovered uh, new fungal orders, family, and species. So specifically, she discovered a fungal order, Mycetalis. I'm sorry, Dr. Hibas, if I'm butchering like these terms. She also discovered like a fungal family, as well as 6 species of yeast and 12 species of molds. As of today, Dr. Hibas has actually published 84 peer-reviewed articles in mycology. And we don't stop there. So Dr. Hibas has also contributed to a number of clinical books, from pediatric infectious diseases, manual of clinical microbiology, as well as clinical microbiology procedures handbooks. So that's the caliber of our resource speaker for today, and she's gonna be talking to you about like emerging diseases from the fungal world. Dr. Hibas?
Hello, hello.
Hello.
Hey, good morning. Maayong buntag kaninyong tanan. Magandang umaga din.
Yes.
Meron ba mga Bisaya at saka Tagalog on eh. And I would like
Congratulations.
to acknowledge the presence of Dr. Dangana. She is with us at the University of Texas Health. Thank you for coming, Dr. Dangana. I know you're there. I saw your name. Anyway, so let's start. I'm gonna share my slide, Dr. Jeff.
Yes, yes.
Okay, should I no. Is it there?
Oh, not yet, Mama.
Oh,
Yeah.
share.
Yes.
Okay. Wait.
You
I'm not
click
in the
one on your keyboard.
I'm in
One,
the middle.
yeah, one and then enter.
Sorry. Okay. So I should see on my slide. I will be talking about fungi as a growing threat to human health today. Sorry, I have a sore throat. And I will also talk about why these fungal infections are emerging now, or are they really emerging? Because I think that they've been there all the time. We just don't see them, or nobody's reporting them. And when nobody's reporting, then out of mind, out of sight, out of mind, right? So, and then also, what fungal infections are fatal? Like those are the dangerous ones. And I will also show you how they look like in humans when they do infections, okay? What do these infections look like in the human body? Okay, so fungi is on the news. There has been several outlets most recently that have been reporting on infection. So for example, CNN said there was a report of the first US cases of treatment-resistant ringworm. So yun yung mga buni na found in patients in New York City. So resistant, so meaning they're hard to treat. And then NDTV said there was a rising cases of mucormycosis or black fungus that is threatening the lives of immunocompromised patients. And then also in NBC News says there was a rare infection of, hold on, rare fungal infection popping up in the unexpected part in the US. And this was about the blastomyces dermatitis infection in the Midwest. So before we go to our formal talk, let's do fungal trivia. So here's a species of fungus. It's called the zombie fungus. And the name of this species is Ophiocordyceps unilateralis. Okay. So what you see here is this fungus is infecting an ant and or insects. They can infect insects. And what you see there are not horns. They are actually fungal structures. They're called cinema. So cinema is a structure of produced by fungus, some fungus species, that is an aggregation of hyphae to form a stalk. So this structure is produced for their dispersal so that they are able to spread and then continue their life cycle. So what happens here is that this zombie fungus they are infecting the ant, and they live there as a parasitic symbiont, meaning that they're able to live in the host, but eventually it will kill the host. So because sometimes they travel to the brain, and when they produce the horns like that, or the cinema, then the host will be killed. And then another article was also showing some ants with infection of this fungus as well in a tropical forest. And this was in 2011. So as you see there, they're looking like horns, but they're actually the cinema of the fungus. So I mentioned this fungus because they have been in a lead role in a TV series most recently, or a TV movie, I think. So who has seen The Last of Us? Has anybody heard of this TV movie? This is supposed to be a post-apocalyptic drama television series about a fungus pandemic. So do you think, they said it's unlikely but not impossible, but what do you think? I think, so here, what happened here is that people were infected with this fungus and they become zombies. So when they become zombies, they look like this. So you see those, they look like mushrooms that are on trees, but they are human beings transformed into this kind of mushrooms. But so what do you think? Do you think this can happen to us? Well, I don't think so. Why? Because Ophiocordyceps cannot survive in the human thermal environment. So we have our temperature is 37, right? And they are cold-blooded, so they can only infect insects which are below 37 degrees centigrade. And also, the human body is so complex that its immune system is very their immune hold on. Their immune system would act as a deterrent for this fungus to survive. So I don't think this will happen. But one thing did happen back in the 10th century. So this was a condition called St. Anthony's fire, or another name for this was ergotism. And this was caused by another fungus called Claviceps purpurea. So this fungus is a plant pathogen, and they infect the wheat and rye, and then contaminates the plants with toxic alkaloids. So when this happens, so when they harvest the wheat and the rye, then the flour that is made from the wheat and the rye is contaminated with this ergot alkaloids. And so when the people consume the flour or the bread made with flour, contaminated flour, then they would feel these effects. So they would have dramatic constriction of blood vessels, that the flow of blood to limb is completely cut off, causing the burning and gangrene formation. So they can either be mutilated or they can die. Also, the ergot alkaloids can cause hallucinations, convulsions, and delirium. So this happened in the 10th century. So around 60,000 people died from what they call the Holy Fire in France. So because of this, there were a lot of women who were accused of being witches. So they were burned at stakes because they said, oh, these women are witches and they caused the holy fire. So that was a story. But nowadays, we don't see that most of the time, because the Claviceps purpurea has been prevented from infecting plants nowadays. So that's true. And it's not like the zombie fungus. Okay, so before we go into our main talk, I'd like to just have foundational stuff or fact here today. So there are two growth forms of fungi, okay? So one is a yeast and the other is a mold. So if you notice, the mold is spelled M-O-U-L-D instead of M-O-L-D. So the mold, M-O-U-L-D, is a British spelling, but we in the fungus community and the mycology community, we would like to use M-O-U-L-D to distinguish it from mold, M-O-L-D, because mold can also be defined as something that would give shape to another thing, okay? So if we're talking about mold, M-O-U-L-D, then we know that we are talking about fungi. So, but here in America, in the US, there's been a lot of resistance. So they would rather have M-O-L-D instead of M-O-U-L-D. So that's that. Because you might wonder why you're seeing two different spellings. Okay, so what is the infection caused by fungus called? It's called mycosis. And so there was an article published from our lab by our director, Dr. Wiederhold, and he talked about emerging fungal infections and what are the potential underlying causes of emergence. Okay, so he discussed several species, why these are emerging. So I summarized them here. So the emergence would be associated with underlying conditions like SARS-CoV-2 or COVID-19 infection. So as you heard, I mean, you probably saw some articles when there was the COVID surge, there were a lot of reports about the black fungus. So the black fungus is caused by Mucorales or species like Rhizopus and Mucor. And this came after a person has been infected with COVID and treated with COVID. So they became immune compromised. So then the mucormycosis came in. And then also another cause of emergence would be the topical use of products containing corticosteroids, sorry, spelling, in antifungals. And also poorly controlled diabetes. So diabetes mellitus would be a precondition for fungal infection, we find. And then also there's the environmental exposure of these normally growing fungi in the environment to azole-like antifungals that are just put out there in nature, what do you call that? When hospitals or companies, they just throw their garbage with these antifungals in the environment, then the fungi that are there are exposed and then they become resistant. And because of that, then when they infect some patients, then the clinicians would notice that there are some resistant fungi that are causing infection, and so they become aware of this infection. So that will be causing emergence as well. And so also antifungal resistance, and then it leads to awareness of a fungal organism that's causing this that is causing the infection that is resistant to antifungal treatment. Okay, other underlying causes of emergence would be also the use of azoles in clinical settings, because most of the time, azoles would probably work than other antifungals. So the increased use could also lead to resistance. And also there's the inadequate infection control in healthcare settings, in hospitals with patients with chronic medical conditions. So that would also cause emergence of fungal infection. And then there's the increased awareness of antifungal susceptibility testing and surveillance. Because of these growing or rising cases of resistant species or strains, then more and more clinicians are aware of these organisms, of these species, and that's how they are said to be emerging. And then also there's the improved methods of fungal species identification. When before there was the infection, and then because they just say, oh, it's a fungal infection, we treat it. But because now we can now identify it to the species level, because we have improved methods using DNA sequencing, then we know what's causing the infection. And so we know what fungal species are emerging to cause these infections. And then also climate change. So for some fungi species that normally lives in a colder climates, we know that with climate change, there's warming. So when there's warming, then these fungal species could adapt to the warm environment. And our bodies are warm. So when they do that, when they are adapted to the warm environment, then they have the possibility to be able to cause infection in the human body. Okay. So then in 2022, the WHO published a list of fungal priority pathogens. This is to guide researchers to develop and do whatever action they need to do to be able to prevent infection, because fungal infections are not easy to treat. So this is what they did in 2022. And then somebody did an op-ed, and they did the list. And so this is the list from the WHO. Okay, so Cryptococcus, Candida, Aspergillus, and other species. And then there's a critical priority, there's high priority, and there's medium priority groups. Okay, but somebody did the list again, and they called it the most dangerous fungi we need to study according to the WHO. But I don't think most dangerous fungi, because there's a lot of fungi and they can be equally dangerous. But yeah, let's go with this list. So the critical priority. So there's 3 species here of yeasts, and then there's the Aspergillus fumigatus. And if you notice, the mortality rate for these strains or species are ranging from 50 to 61%. And that's very, very high. And if you look at their distribution, they're globally distributed, meaning that they're found all over the world. So they can be found in the Philippines as well. So be careful, okay, over there. And then we have the high priority, and these are other yeast species. And the same thing, their mortality rate is from 45 to 60%. And that's really, really high. And also they're globally distributed. And then we have the molds. So we have histoplasma, and usually this is prevalent with people having HIV/AIDS. And we have the mucorales, and you look at the mortality rate, it goes up to 80%. So in the lab, when we receive samples with mucorales species, this is for us considered a fungal emergency. So we need to really do our identification and whatever testing we have to do very fast, so we can give the results right away to the clinicians, so they can treat the patient carefully, correctly with the species that we have given them. And then there's also Fusarium, which has also an equally high mortality rate. Now, mucorales and Fusarium have a high mortality rate because they can cause blood infection. They can go into your bloodstream and cause fungemia. So once they're there, it's very easy. It's very, very difficult to treat, and you can die from it. And then we have the causative agents for Eumycetoma. So the causative agents for this condition is not fatal. Very low mortality, but it has a very, very high social impact. So when you have this disease, some people, they get ostracized. Some people, they live away from their families because of their conditions. They are shamed by what they have. And we will see that later on in the talk. So let's start with the yeasts. Okay, so there's a yeast infection. It's called candidiasis. Okay, the common causes of invasive candidiasis are mostly Candida albicans. So the most common species is Candida albicans. And candidiasis is associated with very high morbidity and mortality because they can cause fungemia or infection of the blood, or they can be invasive, so they can infect the whole body. So these are just phases of candidiasis. So they can be in your folds in your body. If you've seen before, you have babies with some white stuff on their tongue. In Tagalog or maybe in Bisaya, it's called pipita. So that's a condition called candidiasis. So yes, it looks like this. In other phases, they can also infect the nail, and of course, the folds in our body, in our buttocks, in the groin, so wherever, in between your fingers. So this is what candidiasis look like. Other non-Candida albicans species are listed here. So there's Candida glabrata complex, there's Candida crozii, and there's paracillosis, beta-psilosis. So this is a complex. So all these other species of Candida are also equally have high mortality rate as well. So let's look at the Candida glabrata complex. So the Candida glabrata complex. So this is complex. This is a complex because it is comprised of 3 species. And Candida glabrata is part of our gut flora. So sa tiyan, sa intestine natin. So it is a normal flora in our normal microbiome in our gut flora. So, but when they are transmitted to us, not from the gut flora, but by nosocomial transmission, they cause systemic infection. So what's nosocomial infection? This is an infection that you acquire in the hospital. So for those who have children, if you're going to visit somebody in the hospital, you don't need to bring your children into the hospital, okay? Because they might get these nosocomial transmissions or infections. So, and you know how difficult they are to treat. So just be careful. And then also Candida glabrata has been reported to cause 14 to 15% of the infection by nosocomial transmission. And the infection also has been noted to have the highest mortality rate because they are able to produce biofilms. So biofilms is actually a virulent factor for Candida glabrata. And also they also have antifungal resistance to a lot of antifungals. So biofilms is like a mucousy slimy matrix that envelopes the Candida glabrata. So that's why when there's disinfection going on at the hospital, they don't die. So they're still there because of the biofilms that are coating them. So let's look at a case report. So this was a case report by Ferreira et al. This was in 2021. So there was a report of an unusual invasive candidiasis caused by Candida glabrata. So this patient had another condition called systemic lupus erythematosus. So there was a high rate of similarity between the colonizers and the infectious agents. So when we say colonizers, these are your host microbiome. They're the Candida glabrata that's living in your gut, okay? And then the infectious agent is the same, which is also the Candida glabrata. So they were thinking, oh, okay, are these Candida glabrata coming from the microbiome, from the gut? So what they did was, okay, they found that the patient had lupus, and the lupus is associated with host microbiome interaction. So what this does is that it leads to dysbiosis. So in Tagalog, nabubulabog yung host, yung gut microbiome. So when there was so it led to dysbiosis, and because of this, there was dysregulation of the immune system. And then this led to the translocation of the microbiome, so your gut microbiome, into the bloodstream. So when that happened, then you have candidemia, which is a blood infection and so hard to treat, and it goes all over your body and then led to death. So unfortunately, this patient died. So this was a fatal case of invasive candidiasis. So be very careful. Don't bring your children to the hospital to visit somebody, okay? Because you might have those biofilms lying around, and you touch whatever it is, then you might have an infection. And then we'll talk about Candida auris. Now, sorry. Candida auris has also been on the news recently. So it says here, NBC News said deadly fungal infection spreading at an alarming rate in the US. And then CNN said an emerging fungal threat spread at alarming rate in US healthcare facilities. So healthcare facilities, meaning in hospitals, in clinics. And then also ABC News said the growing threat of fungal infections. And this was all about Candida auris. Okay. So Candida auris was described back in 2009 in Japan. So back in Asia, but it quickly spread to 47 countries. And now it's considered endemic in South Africa and India. So the fatality rate among those with C. auris infection, candidemia, is 60%. That's very, very high. So Candida auris has 5 geographical distribution. So we have clade 1, meaning a group of species that are found there. So South Asian clade have been found to have fluconazole resistance. So these are their antifungal susceptibility patterns. So they found. And clade 2, which is East Asian, so this is India and the region. So they found that these strains in East Asia are susceptible to antifungals. So that's good. I mean, you can treat it. But the African clade is also resistant to fluconazole. But for the South American and the Iranian clade, there's no study yet to see what their antifungal susceptibility patterns are. So we don't have any data for this. So, but yeah, these are the 5 geographical distribution for Candida auris. So what are the likely contributors to Candida auris transmission? So what caused it to be spread around? One is the improper use of personal protective equipment. So PPE, your gloves, your lab coat, and then improper disinfection of shared medical equipment and hand hygiene. So it boils down to inadequate healthcare disinfection, and of course, your personal protective equipment. So for example, if you have lab gowns, don't bring it to the house, okay? But maybe you can launder it somewhere else and then before you bring it to your home. And then we go to the molds. So we have, they're not actually medium priority. They're still highly fatal. The mortality rate is still very high, from 53 to 80%. And so we have histoplasma, the mucorales, and fusarium. So let's talk about histoplasma. So histoplasma is one of those species that we call are thermally dimorphic. So what does that mean? thermally meaning high temperature, dimorphic meaning they have two forms. So histoplasma at room temperature is a mold. But when you grow it at high temperature, it becomes a yeast. So if you look at this red, this is called a blood agar, so you can see it's growing like a yeast. But in my other images, they're growing like molds. Okay. So that's why they're called thermally dimorphic. They change their form when they're grown at a higher temperature. So when they're living in the human body, which is 37, they become yeast. And so that's why they can easily invade our human body because they can be transferred from one place to another. So here's a case of disseminated histoplasmosis in the America. Oh, not a case, but they found that histoplasmosis in the Americas is a lethal blind spot. What does that mean? So histoplasma is endemic in the Americas. Sorry. Endemic meaning it's found here in the US, in South America as well. And so most hospitals have no access to rapid diagnostic techniques. So because of that, they won't know that they have histoplasmosis. And because they don't know what the infection is, the clinicians or the doctors cannot stop their patients from dying. Okay. So the public and international health authorities appear to have a blind spot for histoplasmosis. They do not know when it's there. So that's why it's really, really important to have our healthcare facilities to have the ability to diagnose this. And I think histoplasma is also present in the Philippines. I don't know. I think there was one case that I've read somewhere, but I can't remember when it was. But so this is how histoplasmosis look like in human infection. Okay, so we go to mucormycosis caused by species in the mucorales. So as I've said, when you have this infection, it's considered a fungal emergency because these species are very refractory, meaning that they are very, very resistant to antifungal drugs. So one of the example species of the mucorales are Rhizopus and mucor, as pictured here. So we had a study. So in our lab, we have been receiving a lot of samples in the mucorales since the 2015 to 2020. So we did a study on epidemiology and antifungal susceptibilities in the United States. So we had around 854 total number of isolates, which this study represented one of the biggest or the largest epidemiologic and in vitro susceptibility studies of the mucorales. So what did we find? So we found that the highest infection was caused by Rhizopus, as you see on the the blue pie in there. And the next one is mucor. And as you see, these two species are very, very deadly. And the sites of infection are found. So mucor is found mostly in all places in the human body. I mean, not mucor, but Rhizopus. And mucor, which is the orange pie there, is found in the blood. So mucor is very deadly because it can cause fungemia. So once you have that, I don't know, you get lucky if you survive. So then we have a case. Look at a case. So there was an article in 2021, and this was during the surge of COVID. And this article was about a case of rhinocerebral mucormycosis as a sequelae of COVID-19 treatment in India. So sequelae means as a consequence of COVID-19 treatment. Okay, so the reports were primarily from India. And so mucormycosis was called the black fungus, not because they're black, but because when they infect the human body, you would see some blackening of like necrotic tissues around. And it afflicted a number of patients with active SARS-CoV-2 infection or COVID-19. And also there was an increasing number of diabetic patients afflicted with mucor infections. And also India was known to have the highest burden of mucor infection worldwide. So now these factors, in addition to the recent rise of COVID, has led to the rapid increase in the emergence of mucor cases in India. So this is a patient. So as you see, his eye has been infected with mucor. So they were getting him ready for orbital exenteration, meaning that they're going to take out the whole eye because the mucor is inside there and they want to take it out and try to remove the fungal infection inside the eye. So then they were able to take out the whole eye. And then what they did was they need something to plug the hole in the eye. So they took some tissue from the thigh and they put that in the hole where the eye was. And so this was a happy ending. So the patient survived, although of course you see that it's not a very good picture, but hey, she's alive, he is alive, so that's good. Okay, so then there's another case of mucormycosis. So this was caused by leukemia and with an additional condition, an infection caused by Candida albicans. And this one had a fatal outcome. So the story was there was a very healthy 38-year-old male, and he had a bike accident. So he had multiple blunt abdominal and thoracic injuries. So serial rib fractures. So there were right-sided kidney, liver, and biliary tract ruptures. So what they did was they had repeated abdominal surgeries requiring the abdomen to be kept open. So they had to keep the abdomen open because they had to do a lot of surgeries inside. So then the patient developed several syndromes, a syndrome. So they had elevated intra-abdominal pressure due to retroperitoneal hematoma and the swelling of the intra-abdominal organs. And then at day 10, the C. albicans caused an infection in the trachea. At day 12, both the C. albicans, so that's Candida albicans and Leukemia corymbifera, were isolated from a swab. And then, so what they did was they did a rinse of amphotericin B, which is an antifungal. And then day 14, they did another ampho B IV from 40, and then they increased it to 80 milligrams per day with another antifungal F5C. And so at day 16, they took a picture of the open wound in the abdomen, and you can see the fungus growing along the rim of the wound. That's the white stuff that you see in there in the image. So it's actively growing like leukemia. So unfortunately, of course, the patient died, and I was sad because as you know, like leukemia also is very, very refractory and very resistant to antifungals. So they were not able to save the patient. And then we go to fusariosis. So there was a review in 2020 about human fusariosis, which they said is an emerging infection that is very, very also difficult to treat. So this is what fusarium looks like. So in this case, this is a Fusarium solani species complex. So it's growing like white cottony in the agar plate. And then when you're looking at the microscope, they have this boat-shaped canidia or spore. And they're pretty, but they're deadly. Okay. So what are the different spectrum of fusariosis in the human? So in a human infection. So they cause mycotic keratitis. So from contact lens cleaner. So be careful if you're wearing contact lenses. Your cleaner should not be contaminated, so your fluid, the cleaner, because it can cause mycotic keratitis, and you're going to lose your cornea. And then you have endophthalmitis, sinusitis, brain abscess, cystitis, cutaneous infections, and also nail infections, which is what we also call onychomycosis. And so the most prevalent infections are the nail infection, which is onychomycosis, and skin infection, and the mycotic infection in the eye. And the most susceptible people are those with blood cancers, so immunocompromised because of blood cancers. Okay, so we're going to go to agents of Eumycetoma. Agents meaning that those fungi that cause Eumycetoma. So as I've said, their mortality is low, mortality rate is low, but they have very high social impact. And these are also globally distributed. So what is Eumycetoma? So Eumycetoma is a chronic deep fungal infection of the skin and subcutaneous tissue with a protracted disease course if neglected. So protracted means long. So they are endemic in tropical and subtropical countries, that is Africa and Southeast Asia, right where the Philippines is. Okay, so be careful. And then, so the etiologic agents, or those that are causing Eumycetoma, can be fungi or bacteria. And the most common site of infection is the foot and the hands. So how do they look like when you have Eumycetoma? So what we call clinical presentation. So they would appear as plaques with hard, woody swelling. And then they are purulent material discharge. They would have purulent material discharge from sinuses and fistulae. And then there's a presence of characteristic granular grains or microcolonies. So granular grains are those like little beads, parang sago. So they can be white or black. So it looks like sago, okay? There's pearl in the bubble teas. So those ones. And then they are scarring and hyperpigmented in surrounding skin. Okay. So pathogenesis, how do they become a disease? Okay, so the natural habitat of agents of Eumycetoma are, for example, feces or in the soil. And then they are infected. So the host is infected by inoculation. So, excuse me, infection by trauma, or trauma because when probably you walked into the woods or in forest without footwear, so barefoot, and then you get pricked by a thorn or something. And also you can get bitten by an intermediate host. So when that happens, then you could be inoculated with the causative agent that was found in the soil or in feces. Also, it also depends on whether your immune system is strong or immune compromised, or you're immune compromised. So when that happens, when you have the infection, subcutaneous mycosis develops over the course of months to years. So it will start small and then you ignore it, just goes on and on and on. And then before you know it, it becomes it becomes worse, and then you're not able to do anything about it. And then so it it goes untreated, and then it's not very good to look at. So that's how it looks like. So don't ignore it when you have something like I don't know how to call it in Tagalog, but in Visaya it's called hubag, or like little ulcer, something like that. So yes, it will look like this. Of course, you're going to be ashamed to go around. So that's why it has a very high social impact, because you're going to be probably hiding somewhere, not be social anymore because of what you have, and people will not go to you anymore, will shun you. So what happens here, if left untreated, the infection can track down to fascial planes and then eventually will involve the muscle and the bone. And then it leads to complications such as osteomyelitis, and then making it more resistant to treatment. So yes, be very careful and do not ignore anything that would start as a small wound or small infection. So just, and then sometimes you say, okay, I've been treating this for a long time. Why is it not healing? So go see a doctor and see what you have, because you might have eumycetoma. Okay, so how do you evaluate if you have that or not? So punch biopsy is usually done. This is the most specific test, or histopathological examination with skin tissue. And so also examination of grains, those sago-like things and granules. So, and then because you're going to be seeing mycelia, fungal mycelia or fungal, it can grow if you put that on agar. Other methods would be imaging like X-ray, ultrasound, CT scan, and MRI. So etiology. So what causes eumycetoma? So the agents are classified based on grains produced. So there's black grains, white grains, yellow grains, or white or pale unstained grains. So the black grains would be caused by several species, which is Majorella mycetomatis, and then other species like Dermatosphaeria, Exophylla, Medicopsis, Falciformispora. So these are all tongue twisters. So never mind about pronouncing them, just read them. And then Curvularia lunata. Okay, so let's talk about Majorella mycetomatis. Majorella would look like this in culture. So they're pretty, right? So they grow at 37. And then when you look at them in the microscope, they don't produce anything but mycelia and some chlamydospores that are not actually spores. So they cause this kind of mycetoma with multiple discharging sinuses. So what it means that the discharging sinuses would be discharging those balls of mycelia called grains, and these are like the sago-like things there, and it's very ugly to look at. And sometimes they got, so in this case, this was treated for a period of time, for a long period of time, with an antifungal called itraconazole. And then also they had surgery, but it came back, it recurred. So, and then it looked like this. So it's not really pretty. So yeah, you'd rather not be infected. So be very careful when you're walking in the woods. Wear protective clothing, okay? Shoes, whatever, but don't go there barefoot. Okay, so this is another lesion caused by another species called Majorella fahalii. So then we have other species like Exophylla that would also cause the black grain mycetoma, Medicopsis romiroi. So these are what they look like. And Falciformispora. So these are just, so Eumycetoma is caused by different species. Curvularia, acrimonium. So in this case, there was an eumycetoma in the eye, just sitting on your cornea, on the cornea. So what they did here is they tried to take out the mass that is sitting on top of the cornea. And so as you see in the pictures, and then in the end, they were able to take out the mass, and then they did keratoplasty, and then they were able to, I think, save the cornea after all. So yes, very, very ugly infection. And this is Fusarium solani that is causing the same way. So see the bulge in there, it hasn't opened yet, so the grains are inside as seen in the x-ray, so you can see those. And eumycetoma caused by Aspergillus. Who knew? So we've always thought that eumycetoma is caused by Majorella, but these different species that are causing these disease. Scedosporium boydii is also causing eumycetoma. And then this one we call an accident because Microsporum canis is a skin infecting fungal pathogen. So this is the one that would cause your buni, your an. So that's what it does. But in this case, I think there was a breach in the follicles. So that's why the microsporum, excuse me, the Microsporum canis went underneath the subcutaneous layer, and so they caused that's why we call it a pseudomycetoma, because it's not really real eumycetoma, but it probably an accident. Okay, but it got healed. So after a year of 2 years, it says here, of treatment with griseofulvin. That's a long time of treatment. So what I've shown you with the eumycetoma is that it is very, very important to know what species is causing it, because if you knew it was Majorella, then you would know what would work with Majorella, okay? But if you knew it was Aspergillus, maybe you would do one that would work with Aspergillus like voriconazole. So that's why nowadays people would just say, okay, that's a fungal infection, let's treat it. They would want to know what the species is that is causing the infection, so that they can really hone into what the proper antifungal to use for treatment, because as you know, antifungal drugs are very toxic as well. So you cannot just treat and treat and treat, because eventually it will cause toxicity, hepatic toxicity. So it can damage your liver. So either you will live because you were able to treat your mycosis, or you will die because you had hepatotoxicity. So there's that. And so that's why we want to really know what this fungus species is, so you can do proper treatment for that infection. So other mycosis. So here there was a case report in Brazil. There was a 65-year-old farmer in Ceará. So in this story, there was a small lesion, like a nodule, parang pimple, like a pimple. So the guy had no symptoms, but gradually the small nodule extended along the leg and the knee, so it spread. And so after 12 months of after the symptoms appeared, that's when he went to treat it. He went to the doctor and said, okay, they treated it with neomycin, but there was no improvement observed. So he ignored it, or he just didn't go back to the doctor, maybe because he didn't have any way to have it treated. For 11 years, it festered. So he went back to the doctor again after 11 years and reported the appearance of varicose lesions that started on the back and outer side of the right foot. So when he went to the doctor, it was already like this. So it started with the just pimple-like nodule on the foot. And then at this stage, it was already foul smelling. So there was a foul odor emanating from the lesions. So this guy had to stay away from the family because, well, he couldn't stay because everybody was like, you're so stinky, get away from me. So here, what happened? So what they did was they did all the blood work and they all looked normal. And he was negative for hepatitis and syphilis and HIV. But when they did direct exam from the lesion, they found the presence of muriform cells. So when this happens, when you see these copper bodies, we call them copper bodies. And then this would indicate a certain kind of infection caused by pheohyphomycosis. I mean, pheoid fungi, meaning that these are dark colored fungi. Okay, so the muriform cells would indicate that. So they were able to isolate the fungus and let it and get it to grow, and they were able to identify it as Fungi pedrusoi. So this species is very prevalent in Brazil. And so this is how it looks like. It's so pretty, but that leg is not pretty at all. So what they did was they had a combination of treatment. So there was a combination A, and so which is a combination of an antifungal drug with glucan and imiquimod, which is an immunomodulatory drug. And then there was another combination, so with thioconazole and acetretin. And so acetretin, because when you have this kind of mycetoma or pheohyphomycosis, there's a keratinization. So there's a thickening of the wall around it. So acetretin would inhibit the excessive cell growth. So that the antifungal can go through and penetrate the wound to actually get in contact with the causative agent or the fungus. So what happened? So after 5 months of treatment, this is now what it looked like. So his leg healed. So there's not this. This is so much better looking, and also he was able to return to his family and social life. So yes, if you do have those kinds of infection, just be aware that these things can happen to you, okay? Here's another one caused by Fungi pedrusoi. Again, it took months to years before the patient went to the doctor, and eventually they got healed. So, but yeah, it looks really horrible. And here's another case of an infection caused by Phyllophora verrucosa. See that? Ooh, this was a farmer, and he did not recall any trauma at all. So trauma meaning he was not pricked or whatever it is that was able to inject the causative agent into the skin. But anyway, he developed an infection caused by this species called Phyllophora verrucosa, and they were able to treat this infection, and he got healed. So that lesion disappeared after the treatment. And then we have the Trichophyton mentagrophytes genotype 7. This was an article published in 2023, last year. And this was genotype 7 infection was reported among men who have sex with men. So the transmission of the dermatophytes, especially Trichophyton mentagrophytes genotype 7, was through sexual intercourse. So there were 13 cases in France, and 12 of these men had sex with men, and some of them traveled to Southeast Asia. I think it was Thailand, and came back to France, and then they spread the infection by having sex with other men. So this is now trending, and 2 months ago I found an isolate submitted to us that was also a sexual from a patient with sex with other men. So we had the first case of genotype 7 here in the US. So this is how it looks like. So you would have those nodules on the beard area, and also on around the anus. So there's a lot of those papules around the puet. So yes, and also this is resistant to terbinafine. So thank you so much for listening. So hopefully you're able to eat your lunch today after seeing all that, okay? Okay, Jeff,
All right. So,
if there
thank
are
you so
questions.
much. Thank you so much, Dr. Hiba. So I believe there's actually like one question
Okay.
from Zoom po, and it's from Joel Pagaduan. So it's actually more a question po of coconut oil or VCO, can it be used as antifungal?
I don't know. Has there been studies in the Philippines for that? I know there's a lot of VCO products there, right?
Yep,
I don't know if they have antifungal properties.
I
Would
see.
you know? Like I don't know. Honestly.
I also wouldn't know din po. Yeah, because I bet, like, siguro po there are like some products being shared out in the market claiming antifungal property, but like it's like we're not experts to speculate
No.
as well. Okay. Um, Question po, it just came in from Sir John Carlo Tanzo. So, good morning, doctor. I'm a senior high school teacher and planning to conduct fungi viewing at my bio class. I would like to inquire about the safety of observing fungi through potassium hydroxide in a school setting, considering the presence of fungal spores even when the specimen is derived from the human body, such as dandruff and others.
um for me, because I'm a clinical mycologist, I would recommend it because with
You wouldn't, you would,
I
you,
wouldn't I
yeah.
wouldn't recommend it. Maybe just use videos unless you have containment in the school. So maybe you can bring them to a lab where they can observe these things, but in classroom, I wouldn't recommend.
And Dr. Hibas, I know that this question just keeps coming up. Like, I think even in the past mycology basic training courses that we have provided over the years,
Mhm.
I think it's like it's first like it's good po Sir um what's his name? It's good po Sir John that like you're raising interest rin po in microbiology and also mycology. But then I think based on what we have also learned from Dr. Hibas over the years, that safety is also like really, really important when it comes to fungi because we just wouldn't know if they're pathogenic or not. And if I recall it from your lectures, Dr. Hibas, like the use of a biofume hood is typically advised. And so in the absence of that, there's like so much risk that we won't be able to predict. Is that right, Dr. Hibas?
Yeah, not biofume hood,
And
Dr. F.
yeah.
It's biosafety cabinet.
Oh, biosafety cabinet. All
So
right,
fume
all right.
hood would be for chemicals.
Okay, okay, okay.
Yeah.
All right.
So what was your question?
So yun po. So
Oh yeah.
yeah, if that would be safe. So you said
Yeah.
like you would not if there are options of exposing them through video viewing. Yeah, that would be preferable. Now, a question from Francesca Libunao. Hello po. I want to ask if urbanization can influence the prevalence and distribution of fungal infections in humans.
I don't know. Urbanization meaning getting rid of our natural forests and things like that. Would that be?
Yeah.
Or urbanization like waste disposal not very good. That would be a factor because then, as I said, there was a lot of exposure of the normal naturally occurring fungi in the environment exposed to these chemicals that are leading to antifungal resistance. And then so when they infect people, then they're very hard to treat. And also a lot of people now have underlying conditions. So they even fungi that are not normally dangerous, they're opportunists. So when they get into the body, then they cause infection. So, but urbanization, I don't know which context, in which context are we talking about?
Yeah. And I would like to follow up on that, Dr. Ibas, because that's
Yeah.
actually one of the first things that I picked up from the intro. You mentioned that um, improper disposal of antifungals
Mm,
to the exposed environmental fungi
mm.
that would result in resistance. And so, how important is it for medical facilities to actually handle or to dispose of those antifungals properly?
Very, very important. And as you know, I don't know in the Philippines if they're well policed.
So essentially, there's an inventory of the flow
Is that
of
right?
antifungals in hospitals in the US.
Oh, okay. Are you asking me?
Yeah, I'm asking you. Yeah.
Yeah, yeah.
Okay. So how about po yung because I'm not sure po in the Philippines if there are over the counter antifungals like so does that even mean that okay they should not just like throw it out?
Yes.
Okay.
That's why so for example, here in the US, so I have a couple of medications that I don't need anymore because I'm done. You can't just throw it out in the garbage. So I give it to my doctor and she will be the one to dispose of them. So they have their own disposals. So biosafety again. It's like your biosafety program in the city should be very good. In hospitals or wherever institutions, there should be a program, like a section that would deal with those kinds of wastes.
Okay, okay. Sa mga kababayan po nating Pilipino, so if you're using antifungals at hindi niyo na po kailangan gamitin, huwag niyo pong basta-basta itatapon kasi that could expose fungi that would react to it and would cause them to be resistant. And then when they propagate and infect others, then that medication hindi na po siya gagana. All right. Okay. There's a lot. So I think our audience members are actually getting more and more engaged and triggered to ask questions. So there's a number of questions that came through. So from Sir Ricardo Cordova, Is there an increasing prevalence of fungal infection also in the Philippines? And will it also be a problem here, considering the hot and sunny weather?
Yes, there is, but we don't know because, as you know, fungal infection is not notifiable in the Philippines. So what I mean in not notifiable is that when there's an infection, nobody is reporting it. So only now that people are starting to publish articles about, prevalence and infections around the regions or things like that, but still not enough. So really, there should be some kind of surveillance, but we don't have that. No one is really interested in studying fungi.
Yep. And
Yeah.
so, Dr. Hiba, so I think there could also be the way I understood it from your discussion, and like in light of that question that was asked, is that there seems to also be like a chicken and egg paradox happening here. um I think based on our personal conversations, the landscape in the US is actually well structured such that when patients come to the hospitals, there's a well established sampling mechanism for the fungi to be identified, just so that the treatment is targeted, specific to what that species
Yes.
is. So
Yes.
maybe po, a question that I actually have written this like in my notes earlier as I was listening. Could you give us an idea of like the workflow po? Say for example, I have a friend who has like an infection, and I brought him to the University of Texas San Antonio Hospital. And like, okay, could you look into this? It looks like a fungal infection. What is that process like po? And timeline rin at different stages of identification and then treatment.
Okay. So the patient goes to the hospital and the doctor sees, oh, you probably have a fungal infection. So they will take a sample, so maybe a punch biopsy or a swab or take a tissue and then send it to the lab. So that'll probably take a week before, so they want to grow it. So it will take probably a week or two to grow the fungus. But then when they see a fungus growing, sometimes because they're growing from the human body, so they're also distressed. So they don't grow normally. So they will not produce what structures they should be producing normally. So when that happens, we call the fungus sterile, meaning it doesn't have anything, any structure except hyphae, which is the filament. So there's nothing to go by for identification. So what they do, okay, you have a fungal infection because you've been languishing there for 2 weeks already. So we're going to do an empiric treatment, meaning we treat you by experience. Like, okay, we've done this before, we will give you this. Maybe you will heal or not from this. But then if nothing happens, then they will send the sample to us for identification. So then for us, because it was sterile when it was with them, so we're going to grow them in different culture medium. And then sometimes it will take time. And in this case, because they've been languishing already in that other lab, in that other hospital, so they come to us already a month into the infection, right? So for us, the onus is to give as much to make the turnaround time. So turnaround time meaning when they come to us, we give the result back very fast, like, okay, as fast as we can. So as instead of waiting for the fungus to make structures, we send it for DNA sequencing.
Mm.
So when we do DNA sequencing, it will take a week. So we send it, even if we know that it is sterile, but we look at where the substrate is, where it was isolated. So if it was isolated from the bronchoalveolar lavage. Maybe it's one of those aspergillus something. Or if it was from the skin, then probably we think of dermatophytes like trichophyton. So we look at where it was isolated from, just so we have an idea, even if it was sterile. So we send it for sequencing. It will come back after 1 week. And meantime, the fungus is growing. So when the sequences come in, the fungus is already producing something. So whatever we have as a result of the sequencing, what ID we have, what identification, then we confirm it with the morphology, which is probably already been done by the fungus because it's been growing for a week already. So how long does that take? 2 weeks already.
Yeah, but then what I picked up from your answer, Dr. Hibas, is that so to really confirm it, you resort to, or you leverage DNA sequencing.
Yes,
Which
because that's the fastest that we can get. Instead of waiting for it for 2 weeks or a month to produce a structure so we can identify it, we use DNA sequencing to see, okay, maybe give a presumptive, to get a presumptive identification, like, okay, this looks like an aspergillus. And then we confirm with the morphology that is from our culture.
Okay, so hindi po pala ganun kasimple, no? It's not like
No,
more of like, okay, you just look under the microscope and then you know.
no. And
Okay.
also DNA sequencing, you need to know what genes to sequence.
Yep. Okay, thank you so much, Dr. Hibas. That's actually very insightful. So hopefully po, ano, we're not sure if there's anyone in the audience who's a clinical mycologist or specializing in a related area in the Philippines. But if you know of how it's done back home po, it would be great to share that insight with our fellow audience members. All right. So
There's a question, Jeff.
there's
Oh,
yeah, there's a lot of questions, Mama. Which one? So I'm
Mark Mark Lopez. So I'm just wondering, Dr. Connie, have you come across about incidence rate of fungal disease among those who study fungi? Not really, because we are really very, very careful. We exercise a lot of precaution. We wear gloves. We have lab coats, but we still have the paper lab coat on top of it that is going to be thrown away after we do it. And then there's the biosafety cabinet that we use. So there's many, many precautions that we use. So there's no reason for you to be infected with fungus. Otherwise, when that happens, then something's wrong with your technique,
yep.
that you're not being careful. So no, Mark, we haven't. I don't know anyone who has had fungal infection who is also studying fungi. Oh, and maybe you're talking about when you're doing research in fungal. So yeah, more so. So for example, in the lab, when we have samples from outside, from the environment, we don't accept them because we don't know what is there. We don't know what kind of fungus we have. So usually we don't accept environmental samples because there might be some really dangerous fungi in that sample that we don't know about. So
Okay. And there's a question po that was sent directly to me. Nahihiya po yata yung
why?
a member. Okay, so he said, good day po. I would like to know if overusing antifungal drugs increases the chance of opportunistic pathogens attaining resistance. And,
Yes,
yep.
it's
And then
been
there's
happening. It's happening.
and then there's a couple of follow-ups to that po.
Uh,
How are we going to prevent these? And what are the proper measures that the scientific community should do regarding this matter?
and that's why I really recommend to know, to identify the fungi to species level so that you can have a targeted treatment. You just don't use like, okay, lahat ng antifungals because I don't know which one would work. So, for example, I have an Aspergillus fumigatus. Recently, there's a lot of studies on Aspergillus, and they found that voriconazole would work with Aspergillus fumigatus. So if we know that it's Aspergillus fumigatus, then we don't have to try other antifungals like Ampho-B is very toxic. So we go right away to Vori. Okay. So that's why so before I was always talking about this. So before doctors would look at clinical presentation and say, oh, this is caused by this, or we will treat it like this. But as I've shown you with Eumycetoma, it's not only caused by one organism, by one species. It's caused by different species causing the same infection, the same Eumycetoma. So if you know that it's Majorella, you know you can use a certain antifungal. If you know it's Exophyla, then there's another antifungal that might work. Aspergillus, for example. So that's why nowadays it's really, so as I've said, paradigm shift from just looking at how the infection looks like to what is causing the infection, so that you can have a more specific treatment for that fungus to avoid toxicity and to avoid also uh, you know, to have, what do you call that, a targeted treatment.
Okay. So more and more questions are coming through, but I'm gonna read up a comment from Sir John Carlo, the senior high teacher, who mentioned that he intends to introduce fungal studies like to his students. So he said that, thank you for your response to my inquiry, doctor. In the absence of my level 2 biosafety cabinet, I will utilize visual aids and commercially available slides while adhering to personal protective equipment and laboratory safety regulations. So that's actually very
So
important.
if they can afford to buy slides, that'd be good too.
Mm.
Slides, so made slides by
So,
companies.
okay. All right. Okay, now, more questions. One from Carolina Ampere. You mentioned about Aspergillus niger, Fusarium solani, Rhizopus as threats to human health. I'm a plant pathologist here in the Philippines, and we are dealing with these fungi as common plant pathogens. Please advise us, how can we further protect ourselves against these fungi?
Yeah, Aspergillus niger, Fusarium solani, Rhizopus, these are very, very refractory. So when I say refractory fungi, they're very resistant to almost all antifungals. Walang gumagana. So you just have to be is this Carolina? So yeah, you have to be very careful with handling. So sterile technique, and preventive PPE, very good PPE. And also when you're doing your culture technique in a biosafety cabinet. So here in the US, we don't use the gas Bunsen burners anymore, because what that does is that, so you have a biosafety cabinet, right? So you have a curtain of air that is supposed to protect you from being contaminated by what you're working in, and also protect what you're working inside the biosafety cabinet from being contaminated. So, but when you're using Bunsen burners, it will disrupt that curtain of air.
Air
So,
currents
yeah,
of
because of the thing. So we don't use that anymore. So we use incinerators. Also, if you don't use incinerators, or you can use sterile sticks, so you don't have to flame them. So you don't need the flame. So if you have allowed sterile sticks inside, then you just put them into the like an autoclave container, and then you can autoclave after.
Yep.
And also, yeah, go ahead, Jeff.
Dr. Hibas, I also have like another question po, no? So this is actually handling in the lab. But for people who are going out in the field to sample, um, I can actually relate this to I think a case earlier that you mentioned that like people are getting punctured by like with thorns,
Yeah.
plants that actually have like those spores like
Mm.
on the surface, but like for plant pathologists po siguro who go out in the field to collect samples, how can they actually do it safely?
So proper wear, proper protective clothing.
Okay.
So yes. So from the gown to your footwear and gloves, please. Gloves that are not flimsy.
Okay. All right.
Okay. And also, so Carolina, on top of that, so for Fusarium solani, and you said you're a plant pathologist. We are also seeing a lot of Fusarium oxysporum infection here. So I know that Fusarium oxysporum there is a plant pathogen. I mean, for banana, right? So just be careful. Like usually, just be careful with how you handle those microorganisms.
All right.
Not very cavalier about them being cavalier.
Next, Jonathan Soriano. Good morning, Doc. I am Jonathan Soriano, MS Biology student from Isabela State University, Cabagan campus. I am just curious po about the fungal infection which can be acquired through men-to-men sexual intercourse. So my question po, possible po kaya that this infection can be misinterpreted as HIV or AIDS since the mode of infection is most likely the same? What are the possible treatments po if ever or precautionary measures needed to be done to avoid such infections?
I don't know about being maybe, but your doctor would know whether it's HIV, AIDS or not. So, but for the genotype 7, they can try to isolate it, and then when they see that, then they will know that it's not one. Because if you have HIV, AIDS, you're like a petri dish. You will grow everything. Not only that genotype 7 dermatophyte. So because you're immunocompromised, so they love you. The fungi would love you, and bacteria and whatever viruses you have. So, and on treatment, so that's why this is emerging, because usually treatment for dermatophytes is terbinafine and other antifungals, but this one has been found to be terbinafine resistant. So in the lab, we are trying to study what genes are being expressed when a fungus become resistant to an antifungal. So we found, for example, Aspergillus fumigatus. So we did a lot of whole genome sequencing, and we found that what we call the CYP51 gene is giving the resistance to this Aspergillus fumigatus. So if you know that gene, then you probably can do something about, mutation. Um, what is that method nowadays that being used, that you can change gene,
Chris,
um,
CRISPR?
CRISPR, so you can take out that one and probably change it into something. Well, I don't know that because I'm not a molecular biologist.
Process. Okay. All right.
Yeah.
I'm not sure if this is a comment or a question from Sir Ricardo Cordova. Fungi are opportunistic. During antibiotic use, they usually multiply. How do we go about it?
I don't know about antibiotic because they're not actually affected by antibiotic because um it's probably because the effect of antibiotic to your body. I don't know. Um I cannot answer that question, sorry. Because I don't know if Dr. Dangana is still here. Maybe you can answer that. Dr. Thelma, are you still here? Dr. Dangana is not here now.
All right. Next question is from LV Pro- Procurato. Doc Connie, are dermatologists or could they determine whether an infection is caused by a fungi species or is it just an allergy?
That's the thing. They have to do isolation just to see if it's fungal or not.
Okay.
Yeah.
All right. And then, so I think that's actually like mostly the questions that we got online. We also encourage you all asking questions live if there's anything, if there's any question that you have in mind. And while we're waiting for more questions to come through, um, Dr. Hibas, I actually like one more thing that caught my attention earlier was the idea of biofilm. So I didn't realize that certain fungi actually have the mechanism to form biofilms to essentially protect itself, right?
Yeah.
Like from
They
disinfection.
protected from dehydration. So even it's a dry surface, they're still there.
Yeah. So I would assume that hospitals would have a fairly well established like cleaning procedure to ensure that they get to clean up or get rid of as much of these um species regardless of like whether there are biofilms present or not.
You would think.
Yeah, and that's the intention, right? Like but so Dr. Hibas, like maybe for like common households, um are there like common cleaning agents that our audience members can actually use if they're concerned that maybe I'm actually seeing signs of fungi forming and growing on certain surfaces in their houses or in their
Bleach,
bleach. Okay.
5 to 10% bleach.
Okay. What would bleach be able to get through those biofilms? Okay.
Yeah, I think so.
Okay. Okay. And then do they scrub or do they just soak it? Scrub. Okay. Okay.
And then so also so the Philippines is almost like it's a rainy country now, right?
Yeah.
So in your houses, if you find your wall being covered by black spots, you may have the sick house syndrome. And not only that will cause it will actually not it will cause infection, but it will produce those secondary volatile compounds that are actually toxic to you. So if you find that you're always coughing, you have some headaches or something, then it may be that you have stachybotrys in your house that is producing that. So anything that's black, fungal like on your wall that is always moist, that probably would be. You have to like wipe it out with bleach.
All right. Are there any more questions from the audience? All right. So, okay. So I think like with that, we can actually wrap up like the Q&A portion. So, Dr. Hibas, like any key message to everyone, like given the emergence of fungal infection, right? And like siguro medyo nagulat yung audience members natin, no, when it comes to the pictures that were shared earlier. But then I think it is actually very important as well that uh people are aware of like how serious fungal infections can be. So Dr. Hibas, any message especially to our audience members from the Philippines who may not be aware that hey, this is actually an emerging concern.
Yeah, because as you know, a lot of fungus. So fungi has a lot of species. So we have about 10 to 12 million species, but we only know about 200,000 of them. So we still have a lot to discover. But of that 200,000, we only know about 700 that are real pathogens. So when I say real pathogens, when they get into you, they make you sick. Okay? So true pathogens. But All the rest are opportunists. So meaning normally they're harmless, but because many of us have underlying conditions, then that's the time that they can cause infection. So keep yourself healthy. Try to just clean your house with anything moist or something. Like if there's something wet parts of your house, try to dry it out, just so you don't encourage the growth of fungi around there, because there's a lot of them growing and they're normally growing, and they're so fungi is ubiquitous. They're all around us. But because we more and more, a lot of us are not healthy anymore. So many of our people in the Philippines, it's actually an epidemic of diabetes, right? So that then when you have that, you are prone to a lot of infections, especially fungal infection. So yeah, keep yourself healthy, clean the house, and just be careful.
All right. Thank
Yeah.
you so much, Dr. Hiba. So I think that's it. Like we're not getting more questions. So people are thanking you for this. So they appreciate like the content as usual. They said that they learn new information in all of our webinars. So again, like we were actually able to have about 70 people participating in this session today. This is our 109th event, and one thing that I can promise is that next year we're gonna be back in action. So we're gonna be super active again, and we're intending to uh, uh, book up all of your Saturdays, if not most of them, in 2025. So we have math tutorial sessions that will be launched tomorrow. So if you want to learn more about vectors, if you have friends who are in senior high or in college who would like to learn more about vectors and a refresher on the topic, we actually have a professor from the University of the Philippines, Los Baños, in mathematics, delivering those lectures at least once a month, focusing on the topics that you request. And then we will also have some physics tutorials in line, as well as like STEM pedagogical training courses with some academic partners. And then we're gonna continue our practical research training courses for next year. And then Dr. Hibas also um, mentioned that we are gonna have our 2025 mycology camp. So that's covering fundamentals of mycology all the way to research development and application. So watch out for it next year. Okay, there's just like another topic. I would another con po from Francesca Libuna. I would like to share din po that we have a mycology topic covered in our medical biology course. It helped us a lot to have an initial idea of fungal species. Thank you so much po, Philsci Hub, for a deep dive information for this. So I learned a lot. Yeah, so we'll have a mycology camp next year. All right, I am going to be posting the attendance form. So you can actually fill out the attendance form, and that's how you'll also request like an attendance certificate. So I would just like to encourage everyone to double check the information that you input in the form, kasi whatever is in there will be the information that you'll find in your certificates of attendance. And then we also request that you give Philsci Hub a week or two for us to be able to generate those certificates and send them to you through email. All right. And so with that, we would like to thank all of you for tuning in. And then maybe if you would like, if you're open to it, maybe we can have a group picture po. So dun sa mga game, magpakita ng inyong mga magagandang pa. We'd like to welcome you to turn your cameras on just so that we can all have a group photo. Ayan. Sino pong gusto magpa-photo op kay Dr. Hibas? Ayan. Nag-isa-isa na po sila nagpapakita. All right, so
All right.
we're gonna give people a minute now
Good
for
to
the,
see you all. Hey.
ayan. I know we have a mix of audience. Po siguro we have students, and then we also have practitioners in the fields as well as educators. So maraming salamat po. And nakita ko yung pamangkin nyo, Mama.
Carly is here in the
Ayan.
house.
All right. Okay, yan. So they're starting to show themselves. Wag po kayong mahiya. Ayan. Okay, sige. Okay, count po of 3. So 1, 2, 3. All right. One more. We're gonna take a picture po of the second panel. All right. 1, 2, 3. All right. Okay, and so with that, maraming salamat po sa pagsama ninyo sa amin. So, it's 11:50 in the morning in the Philippines, 10 minutes before siguro mag-start na po ang inyong mga tanghalian. Kita-kits po ulit tayo in upcoming webinars. And again, tomorrow, if you would like to learn more, to refresh your understanding of vectors, we have our first math tutorial session. So with that, maraming salamat po, and you enjoy the rest of your weekend.
Thank you so much, everyone.
Thank you so much po.
Bye, Jeff.